Hi, my name is Greg Gilbert, and I'm a professor here in the Department of Environmental Studies at the University of California, Santa Cruz. I'd like to take an opportunity to tell you a little bit about the research and teaching that I do. How I choose what to study is a combination of aesthetics, curiosity, and where I think I can contribute to solving environmental problems. I think what drives my choices can be divided into what I study, where I study it, and what questions I ask. What I mostly study are trees and fungi. I didn't really choose them, they chose me. I've known I wanted to spend my life working with trees since I was a teenager, when I spent as much time as I could wandering through the forests of the Appalachian Mountains where I grew up. Sometimes it was exploring the hillside for an afternoon, and sometimes it was out backpacking for months at a time. Trees were always magical because they created the structure of the ecosystems on which everything else depended, from insects to birds to the water that drained into the streams where I fished. Even then, I took to heart that the world needs loraxes who speak for the trees. In college, I discovered fungi through courses I took and through some very special teachers. It was actually studying the succession of fungi that grew in a bowl of horse dung in an introductory mycology class, and then discovering a new species of microscopic fungus in that dung that got me really excited about fungi. I was overwhelmed by the tremendous diversity of fungi hidden all around us, and by the important roles that they play as decomposers and pathogens and as normal parts of ecosystems. Plus, under the microscope, the fungi were spectacularly beautiful. One semester as an undergrad, I made hundreds of drawings of fungi through the microscope, which totally drew me into that hyperdiverse world. That led me to plant pathology as a way of studying both fungi and plants and their interactions in the context of significant ecological and economic problems. Where I study plants and fungi has lots of dimensions. Geographically, I split my research time between tropical rainforests and California ecosystems. I lived and worked for many years in the Republic of Panama, studying diseases of tropical forest trees. I've worked continuously there since 1991 as an associate of the Smithsonian Tropical Research Institute, working in highly diverse rainforests, low-diversity mangrove forests, vast stretches of coconut palms, and more recently in the urban forests of Panama City. I've also worked in many other tropical countries, including Costa Rica, Mexico, Colombia, Australia, and Micronesia. I work in the tropics because I'm fascinated by understanding how biological diversity works, and the tropics are the most diverse ecosystems. I work in California because of the tremendous forest and grassland systems in this Mediterranean climate, because of the many disruptive threats to those ecosystems, and because I'm committed to involving as many students as I can in active, experiential, inquiry-based learning in forest and grassland ecosystems. And there's no better place in the world to do that than the Central Coast of California. The questions I ask come from a range of inspirations. Many come from questions that come to me from other people with particular needs. I've helped track down the cause of massive die-off of coconut palms in Cunayala, the semi-autonomous indigenous nation on the Caribbean coast of the coast of Panama, working closely with Cuna collaborators and colleagues from the Ministry of Agriculture. I'm working with the mayor's office in the capital city of Panama to create a robust, ongoing program to assess public risks from diseased trees that can fall on cars, buildings, or people, and to help design management systems that both increase the greenness of Panama City and ensure public safety. With collaborators, I'm working to apply new epidemiological tools to understand and address the spread of a new invasive pathogen spreading quickly through the riparian forests, urban forests, and avocado groves in Southern California. And I'm following my scientific curiosity to see how we can use evolutionary principles and new molecular techniques to understand the networks of interactions between microscopic fungi and the diverse plant communities on the UC Santa Cruz campus. And through the UC Santa Cruz Forest Ecology Research Plot, students and I are following how the coastal forest is changing over time in the face of changing temperatures and precipitation. Some of my work is driven by social need, some by concern over environmental threats, and all of it is driven by curiosity. I'd like to share an example of the kind of research I do in applied evolutionary ecology. We know that all plants are colonized by many fungi. Some of those fungi benefit the plants. Others cause disease. Much of my work is to apply what we know from evolutionary biology and ecology of plant-fungus interactions to try to predict which plant species are likely to be susceptible to attack by particular fungal pathogens. This is useful in evaluating the risk from introduced pathogens or for managing agricultural or forest communities to reduce disease. We know that the fungi that cause disease differ in how specialized they are. Some cause disease on just a single host species, whereas others have very broad host ranges. In any given plant community, most fungi are able to colonize a number of different plant species, but not all species. In the understory of a Panamanian rainforest, we isolated fungi from diseased leaves, then inoculated pure cultures of the fungi onto a diversity of local host plants, usually about 3 dozen species. We then scored for which host plant species developed disease symptoms. There were a couple of species that were highly specialized to only the host from which they were isolated, the orange ones on the left, and one fungus that caused disease on every host onto which it was inoculated, the red one on the right. But most of the fungi were able to cause disease on some subset of the available local plants, but not all of them. The typical fungus was able to cause disease in a little over a quarter of the local plant species. What makes a particular plant suitable for a fungus to attack is determined by a huge complex of physical and chemical traits. Some of those traits need to be present for a fungus to grow, and other traits need to be absent so that growth is not prevented. Different fungi require different combinations of traits to thrive. In some cases, the presence of a single trait might be able to prevent a fungus from growing, but a whole suite of traits together makes the leaf otherwise suitable for growth, or not. Importantly, those traits are not randomly distributed among different plant or fungal species. In many cases, closely related plants are likely to share many similar traits, whereas distantly related plants are more likely to have divergent traits. Closely related oaks in the genus Quercus share many traits. They are similar, but a bit more different from the tan oak, which is in the same family. Madrone, in a different order, shares fewer traits, and coast redwood, which has been evolving independently from angiosperms for a very long time, is even more different. This pattern of more similar traits among more closely related species is known as a phylogenetic signal. Through a series of experimental studies and analysis of data from the U.S. Department of Agriculture, we learned that this also applies to the host ranges of fungal pathogens. A pathogen is much more likely to be able to attack 2 closely related plant species than 2 plant species that are distantly related. We can describe that evolutionary relationship with a mathematical formula that comes from statistical analysis, and then use that formula to predict whether pathogens are likely to be able to jump from one host plant to another. Ingrid Parker and I worked together to see how we could use this to predict ecological outcomes. In a plant community like this one with 6 species, a given plant will receive fungal spores from all its neighbors. Those spores that arrive from close relatives, like D and E are closely related to focal species C, are more likely to be able to succeed and colonize the plant because the similar suite of traits on that close relative will have selected for fungi able to thrive under those conditions. It's less likely to share from the more distantly related species A and B, and spores that arrive from the very distant relatives like F are much less likely to find a suitable host
suite of host traits in host C. This means that a species surrounded by close relatives should be colonized by lots of pathogenic fungi from its neighbors and would likely develop more disease. A monoculture crop is the classic example of a plant being surrounded by very close relatives. If we know how each plant is related to its neighbors, we can then calculate the probability of being attacked by fungi from all the species in its neighborhood. We tested this model in the Great Meadow of the UC Santa Cruz campus. Together with lots of field helpers, we did extensive surveys of the meadow to identify all the plants that were present and how abundant they were, and we built an evolutionary tree so we knew how they were related to each other. We then introduced 44 plant species that had never been grown in California before into the meadow. These were just grown in tubes, and we removed them before they made seeds. Because we knew how each species was related to all the neighboring species in the surrounding meadow, and could predict how much disease they should get using the model we developed earlier, we found that plant species surrounded by a lot of close relatives actually did get a lot of disease, but those plant species that didn't have many close relatives nearby developed very little disease. So here we took some experimental results from Panama, built a statistical model based on evolutionary theory, made ecological predictions for what should happen in a novel situation, and tested it experimentally in the Great Meadow at UC Santa Cruz. Now we're using these same models to predict the spread of introduced pathogens through forest communities and to design polyculture systems in agriculture that protect against disease. This is the general structure of the kind of research I do, combining observations, theory, and experiments to help us predict what will happen in novel conditions.
My work in applying evolutionary ecology to plant disease ecology has two contributions to people and society. First, I'm helping to develop new ways to be able to predict how natural and managed ecosystems respond to different kinds of disturbances, including introduction of novel invasive pathogens and changing conditions associated with climate change. To be useful in building resilience, to restoring damaged ecosystems, to designing more productive agricultural systems, to preventing us from causing more damage through well-intentioned interventions, we need to use all that we understand from ecology and evolutionary biology in a predictive way, not just to explain what the problems are. Second, I work with students and teachers through public schools, UC Santa Cruz, and internationally to build robust, accessible experiences to help develop the next generation of scientists and science-literate residents who will be making choices and leading us forward. I work to instill the idea that science is about developing the ability to recognize and deal creatively with problems we don't even recognize yet, not just learning a lot of accumulated facts. We are all guests of the green plants, and fungi are their most common and important associates. In a world of global change with a changing climate, a rapid reshuffling of biodiversity around the globe, and a dramatic change in land use patterns, how biological communities respond will determine some of the major challenges we face as a society. I hope to contribute both as a researcher and a teacher to addressing some of those challenges. My path to becoming a faculty member at UC Santa Cruz was full of chance encounters and taking unexpected opportunities. I grew up in a small, conservative Pennsylvania Dutch community and was the only person in my family ever to be interested in science. I was the first to go to a four-year college and the only one to go on as an academic. As a teenager, a friend of the family knew of my interest in trees and pointed me to the State University of New York College of Environmental Science and Forestry. It was the only college I applied to, not really knowing much about other options. From there, a series of important mentors shaped my interests, and when I said I was interested in studying fungal ecology, I was told the only way to do that was through plant pathology. I applied to the graduate school as my professors suggested, and ended up in Wisconsin, which at the time had the leading program in plant pathology. There I studied biological control of diseases of soybeans, alfalfa, and ginseng, and I learned two things. First, that I loved thinking about how microorganisms shape biological communities, and second, that I really didn't like working in Midwest agriculture. A visiting graduate student suggested that I take a field course for graduate students taught through the Organization for Tropical Studies in Costa Rica to learn something about biodiversity in the tropics, and so I did. There I got really excited about the tropics, and after the course, wanted to go back to the tropics, and so I contacted Professor Steve Hubble, a famous forest ecologist who was working in Panama, and he worked with me to write grants, which eventually sent me to Panama for 3 years. I stayed for 5, doing research, learning Spanish, learning how to dance salsa and merengue, playing flute with Latin jazz musicians, and teaching at the University of Panama. I only left Panama because I somehow, surprisingly, got a job as a professor at UC Berkeley, where I moved in 1996. There, I started a really important collaboration with Ingrid Parker on the role of plant pathogens and biological invasions, which continued after she became a professor at UC Santa Cruz. Attracted to the collegial, collaborative, student-focused, interdisciplinary program in environmental studies, I was lucky enough to get a faculty position here at UC Santa Cruz, where I moved in 2000. Ingrid and I continue to do much of our research together, including an experiential learning project of jointly raising a son. I continue to spend a couple of months a year in the rainforests of Panama and the rest of my time in Santa Cruz, doing research and teaching and working to create opportunities for students to experience as much field ecology as they can as we learn together about trees and fungi in natural systems.